Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Roslin, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Bobby Allen from Sartorius Steam Biotech.
Hello, everybody. Thank you for joining me today. My name is indeed Bobby Allen, and today we'll be covering best practices in aseptic sampling from stainless process equipment. So we have a fairly aggressive agenda that will cover the what, why, when, and where of aseptic sampling, the different methodologies, or as I like to call it, the evolution of sampling, the different classifications of sample types, the different collection container types that are used, and of course, best practices for sterility, process monitoring, and endotoxin sampling. So let's start off by making sure that we're all on the same page. What exactly is aseptic sampling? Well, it's the means by which we can aseptically withdraw material from the manufacturing process for analysis while not contaminating or altering the sample or the source of the sample itself. Well, why do we aseptically sample? Well, quite simply, it's to ensure that the safety, quality, identity, strength, and purity of the in-process materials are as they have been previously defined. And quite simply, the failure to comply can result in regulatory action. Well, when should we be aseptically sampling? Well, according to the FDA, at the commencement or completion of any significant manufacturing step or after storage for any long period of time. And there are, of course, a host of compendia references we can turn to provide more specific guidelines should we need them. But where exactly does aseptic sampling occur? Well, here we see a very simplified representation of a typical bioprocess, and sampling should occur at every single step along the way. The samples that were taken will vary depending on where in the process they are being taken from. Across the top, you'll see a variety of assays that can be performed throughout the bioprocess steps listed along the left-hand side. The mechanisms used to aseptically extract samples have been evolving over time. The earliest of these can be seen in the upper right and is quite simply a needle and septum. This method requires a side stream from the process, which can represent a considerable loss, not only in product, but in time, as only a single sample can be taken and the side stream pipe needs to be purged, cleaned, and SIP'd before the next sampling event can occur. Valves and bottle assemblies are the next step, and these are simply three-way or block and bleed valves, which are assembled to a bottle and autoclaved prior to being installed on, again, a side stream pipe. And while we certainly improved operator safety by removing needles from the equation, we are still left with a single sampling event from a side stream pipe, which requires, at minimum, an SIP between successive sampling events. The next improvement came in the form of SIP valves, which are used with the same type of bottle assembly seen on the bottom right, and their primary function is to replace the side stream pipe, thereby reducing the amount of product loss. However, you're still limited to a single sampling event and are still required to SIP between them. Then comes the hybrid equipment, which includes both a reusable component as well as single-use elements. The portion of the device which interfaces with equipment, that blue cartridge you see in the upper left, it's reusable, while the lines used to collect the actual liquid samples are independent of each other and single use. So we can now take multiple samples using the same device at multiple time points using a virgin flow path for each. This really defined the very first aseptic sampling devices, or ASDs, and really represent one of the largest steps forward in modern sampling. While significant improvements have been made at each step along the way, all of these technologies still represent considerable risks and limitations, but they were the forefathers of the cutting-edge turnkey aseptic sampling device technologies available to us today. So the Parental Drug Association recognizes the clear benefits of aseptic sampling devices, and the World Health Organization furthers this sentiment by recommending those aseptic sampling devices be fully single-use and disposable. Why? Because it allows one to take advantage of the many benefits of single-use systems within a stainless BioProcess and during one of the most critical and risky operations. So here along the bottom, you'll see some of the examples of the turnkey aseptic sampling devices that are commercially available today. But what exactly is a turnkey aseptic sampling device? Well, it's a self-contained device that has a mechanism to aseptically remove fluid from the vessel. And because the device is a closed system and arrives pre-sterile, only the external product contact surface needs to be steamed in place. And that happens as part of the routine manufacturing operations as it's connected to the empty vessel during the equipment prep. So they must also have multiple and independent sampling pathways. And because each line can access the process fluid independently through the faceplate, it's no longer necessary to steam in place between sampling events. It must also have an appropriate sample collection vessel at the end of each line. And I do emphasize appropriate here, and we'll dive a little deeper into that topic as we go along. And lastly, it must have a means to aseptically remove the sample collection container once it's been filled. All of these attributes provide a closed system, meaning that before, during, and after sampling, external contaminants are not introduced to the process vessel or to the sample itself. So there are a really wide variety of samples taken during BioProcess, and what those samples are taken for is largely defined by where in the process those samples are being taken. So if we take a step back and look at all of those samples, we can see that they actually can be broken down into one or two categories. That's the microbiological and the process monitoring buckets. And these are based on the type of analysis that are being performed on those samples. So regardless of which category or bucket those samples fall in, they all have one thing in common, and that's they must be aseptic to the vessel from which they are being pulled. Microbiological samples have the additional burden of needing to maintain sterility of the sample as well, whereas process monitoring samples need not be sterile for the actual assay. With microbiological and process monitoring buckets in mind, we can then look at the various types of sampling containers for their suitability for each. So microbiological samples must be taken into a closed system, like a bag, bottle, or a tube, in order to maintain their integrity. Process monitoring samples, on the other hand, because they themselves do not need to be sterile, can be taken into any one of the 5 types of collection containers that are available. So when we step back to take a look at best practices for stability, bioburden, and or retention sampling, we know that we fall into the microbiological bucket and that the systems must be closed and pre-sterilized, but that still leaves us with plenty of options. So selecting the most appropriate container will depend on other factors, like, for example, handling preferences for the laboratory technicians. Do they prefer a bag or a bottle? There's also spatial limitations in cold rooms or freezers. Bottles take up more room than bags, but bottles, on the other hand, line up quite neatly on the shelves, whereas bags do not. And what will those storage conditions be? If they're going to be stored at -80, then specialized containers should be used that can withstand those temperatures. And what about the need to control the amount of volume being sampled? When dealing with drug substance or drug product, the volume being sampled is critical to reducing valuable product loss. So when volume accuracy or sample volume control is a major concern, then bags really are a poor choice because they either have no volume demarcation, or if they do, it's nominal. Tubes and bottles are a much better choice because of their discrete and accurate volume markings. Really, if you give an operator, we find that if you give an operator a 50 ml bag and ask them to take a 10 ml sample, you're going to end up with something much closer to 50 ml than you would 10 ml. So as we move on to the best practices for pH and gas analysis sampling, recall that we are in the process monitoring bucket here. So while we cannot contaminate the vessel when extracting the sample itself does not need to be sterile like it would for the microbiological assay. This makes syringes a very attractive collection vessel option because with pH and gas analysis, time from sample to analysis is really important. For example, in cell culture applications, the cells are constantly consuming oxygen and releasing carbon dioxide. And when people often default to bags for taking all of their samples, because really it just doesn't require a lot of thought to put in the bag. But what we see is quite often that results in technicians taking a sample that was provided to them in a bag, then they cut it open to pour out or pull out the sample into a syringe so that it can then be taken over to the instrumentation. This is a waste of a bag and the additional time needed to execute these steps, which in the end may potentially skew the readings because, as we said, cells are constantly utilizing oxygen and giving off O2. So syringes really are preferred here because with gas and pH analysis, the sample can be taken directly to the analyzer. Syringes are low cost and can be also a great cost-saving mechanism because they can be manifolded together or used in conjunction with a Lohr-activated valve to considerably expand the sampling capacity of a single line of your aseptic sampling device. So when we look a little more closely at gas exchange within a sample, we know that it occurs at the fluid interface of air and the container itself, and that the permeability of the container can also have an impact. So when we compare the ever-popular bag to syringes, we can see that the surface area volume ratio for syringes is significantly lower than that of bags. And while all plastics are permeable to gases, the thicker and more rigid plastic of a syringe has a rate-limiting effect on that exchange. So when we compile all of the data and look at the suitability of the various containers for pH and gas analysis, we can see that across all the categories of cost, volume accuracy, surface area volume ratio, time to analysis, and waste reduction, syringes fare significantly better than bags, bottles, and tubes, and therefore are the recommended collection vessel for this type of analysis. This brings us to our last topic, best practices for endotoxin assay. And again, we fall firmly into the microbiological sample bucket and therefore must be in a pre-sterilized and closed system. But now with endotoxin assay, we find that the material compatibility can play a significant role. It is already well understood that the ability to detect and quantify endotoxins can be affected by the sample storage container itself, and that's because those endotoxins have been shown to absorb to the surfaces of those containers. So in order to find the most appropriate material, we ran a compatibility study on the various collection containers that you see listed here, as well as EVA and PE bags. We ran positive product control to ensure that reagents used in the experiment neither enhanced nor inhibited the results, and ensured, of course, that the starting collection containers were endotoxin-free. 3 units from 4 different lots were used for each of the 4 different types of sample collection containers. 3 units were tested at each time point per material, and there were 4 time points with a positive product control run at each. The resulting data was compiled, but as you can see, it's a little difficult to assess in table format, so it was converted to a graph. And I'd also like to point out here that bags were included in this study, but they were excluded from the data set at this point because of their comparatively and relatively high absorption affinity compared to the other 4 materials. So because it was so far out of scope, it was just eliminated from further review here. So when we look at the graph, when the samples from each time point and container type are pooled, we can see that polycarbonate and polystyrene yield the most accurate results when the endotoxin assay is performed within the first hour of the sample being taken. That polystyrene and PETG bottles give more accurate results than polycarbonate beyond 6 hours from the time the sample was taken, and that polycarbonate and polypropylene exhibit continuous drops in recovery over time, and hence PP eliminated from any further evaluation. So, when drawing conclusions based on the pooled sample data alone, polystyrene and PETG look to perform the best. But with no clear winner, which one do we choose? So let's look deeper into the data. as the standard deviation of the average recovery over time. Here, we can see that the standard deviation for these pooled samples across all time points is lowest for PETG, again, making it appear to be one of the better choices. So, is that what we choose? Well, going back to the data, we looked at the differences between the individual sample containers of the very same type at each time point prior to their being pooled, which is what the other data was. So using the standard deviation, here we can see that polycarbonate and or polystyrene collection containers provide more accurate results when the samples are assayed before 6 hours have elapsed, which again is consistent with the earlier results we saw, and that PETG or polystyrene collection containers appear to be the best when the assay is performed anywhere from 1 to 24 hours after the sampling event. So again, no clear winner. What do we choose? Well, we take the step of compiling that data again, and what we see is for the average pooled recovery as well as the standard deviation between collection vessels at each time point, in this weighted table, we can now more clearly see that the overall score for polystyrene is better than that of both polycarbonate and PETG. So, in conclusion, when sampling for sterility and bioburden, any closed container is appropriate, but selecting one best suited for the handling, storage conditions, and volume control required by the process really is the wisest choice. And pre-sterilized syringes are the best choice for pH and gas analysis. They provide the most accurate results due to their low and stable surface area to volume ratio at the lowest cost. And we also know that there is significant change in the amount of endotoxin recovered across most materials between 1 and 6 hours. Therefore, endotoxin samples should be assayed within the first hour of sample extraction using a polystyrene container, and again, due to their high rate of recovery and stability across lots of material and over time. So thank you very much for listening. Here is my full list of references and, of course, my contact information. And I believe now we're to open up the floor to any questions.
Great. Thanks, Bobby. So,
you're welcome.
The first question is, what kind of tank interface options are available?
The tank interface options. So depending on the vendor, you will see that there are a wide variety of options. They range from 3/4 inch TC. There's also 1.5 and 2 inch TC. There's also something called an Ingold fitting, quite often also known under the name of a B-Bron or a safety port, or even 25 millimeter port. It comes under many, many names, but they're available in those configurations as well. So basically, the standard options you would see on most vessels.
Okay. And what are the product contact materials of construction?
So they will vary depending on whose device you're looking at, but generally speaking, there's 3 materials. Platinum cured silicone, stainless steel, 316 stainless steel, and alternatively, many of the aseptic sampling devices out there come with what's called a polyaceramide, which is a type of plastic for the faceplate. And so those are really the only fluid or process fluid contact materials, whatever the faceplate itself or that connection to the tank is comprised of. So there are options.
And what is the maximum number of sterility samples that can be taken with these aseptic sampling devices?
Well, that will depend on the port that you're using the device on, but the largest of these aseptic sampling devices come in a 2-inch tri-clover fitting with up to 9 independent sampling pathways on them. And it's possible to manifold bags, bottles, tubes. You can manifold those collection containers, and you can do up to 5. It's the most I've ever seen with a purge bag. So that would give you 9 lines times 4, up to 45 samples.
And
Sterility
what
samples
about
are available.
Oh.
I'm sorry, can
And
you
what
repeat
about
that?
automated sampling?
So there are a number of devices or instruments on the market that can interface with some of these aseptic sampling devices. And that's really what you need, where the research needs to be put in, is what instrumentation is available to you and what mechanism they need to interface into the tank. As I said, I know that we have set up several different process streams with automation using aseptic sampling devices. So it is possible, and certainly feel free to reach out and contact me, and we can discuss, what it is you're trying to achieve, what instrumentation you have in mind or may already have on hand, and then how we can make that leap into the tank through one of these ASDs.
Okay, well, thank you very much, Bobby.
You're very, very welcome.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.